MARIO-3 was a phase 2 study for first-line triple-negative breast cancer, and it was modeled very similarly to the IMpassion130 trial, looking at nab-paclitaxel plus atezolizumab, but they added a third agent, eganelisib. And eganelisib is a PI3 gamma inhibitor. And what PI3 gammas do, a little bit differently than how we think of PI3 kinase inhibitors, is it reduces immunosuppression, it expands activated T cell populations, and it increases PD-L1 expression. So really asking two questions here. They took both PD-L1 negative and PD-L1 positive patients of can we improve upon what we see for nab-paclitaxel and atezolizumab for those patients that are PD-L1 positive based on the SP142 assay? And can we also extend this therapy to patients that are PD-L1, have PD-L1 negative tumors? So really asking the question of whether the PI3 gamma can make this a more attractive regimen for those patients that have tumors that are PD-L1 negative. It was a small study. There were only 13 patients, and those broke down by 5 patients that were PD-L1 positive and 8 patients that were PD-L1 negative. What we saw was an overall response rate of 100% for those patients that were PD-L1 positive and an overall response rate of 50% for those that were negative. Now, if you recall, on the IMpassion130 trial, the overall response rate in PD-L1 positive patients was 58.9. So small numbers, but a little bit different than that 100%. So really kind of suggesting that this should be explored further to see whether this really is a true finding, that we're able to get more patients to respond with this combination. And we don't have a definite response rate from IMpassion130 for those that were PD-L1 negative, but certainly intrigued since the minority of patients have PD-L1 expression on their tumors to see whether we may be able to extend this therapy to more patients that it's currently indicated for.